Welcome to this explainer. Today, we are diving into what is honestly a genuine high stakes medical detective story. We're tracking a microscopic fugitive, one that has managed to evade our ultimate capture for decades, the HIV virus. For a long time, this pathogen has basically outsmarted our best cellular defenses. But recently, well, some groundbreaking discoveries across entirely different fields of science are finally giving us the blueprints to corner it. So, before we really get into examining the evidence, I want you to consider a seemingly impossible question. What does a rare muscle disease have to do with curing HIV? It sounds completely unrelated, right? I mean, one heavily affects your physical mobility and the other ravages your immune system. But hold on to that thought, because the answer to that exact question is an absolutely incredible twist that we're going to unravel together by the end of our investigation today.
All right, let's kick things off with Act 1, the break-in.
Okay, let's dive into this. If we're going to catch a fugitive, we first need to understand exactly how it operates. How does this highly specialized virus breach our immune system in the first place? Well, it all starts right at the surface of our immune cells, specifically at a receptor called CD4. Researchers have recently managed to minimize and isolate the exact point of contact, which is a huge deal. They pinpointed that HIV's envelope protein, which is known as GP120, latches onto the CD4 receptor domain D1. To be highly specific, it's an incredibly small region, spanning just amino acids 21 to 64. By isolating this tiny sequence, scientists have mapped the precise molecular lock that the virus picks to gain entry into the cell. But here's the thing. Once the breach happens, the virus doesn't just float freely around the cell. It enters wearing its ultimate disguise, the HIV-1 capsid. Think of the capsid as this heavy-duty viral payload protector. Its primary job is to safely transport the fragile viral genome from the cell's outer edge all the way into the command center, the nucleus. And it has to do all of this without triggering any of the cell's internal alarm systems. And this disguise is, well, it's incredibly sophisticated. Some really groundbreaking research by John Meng Liu and a team at Stanford Medicine reveals that the capsid is basically a master shapeshifter. It uses these structural switches to dynamically alter its surface curvature, literally morphing its lattice structure between hexamer and pentamer shapes. This flexibility is brilliant, honestly. It allows the virus to strategically bind to our own cellular host factors, specifically these transport proteins known as NUP153 and CPSF6. So it essentially hijacks our own cellular transport machinery. It coordinates multiple interactions at a single site just to ensure a smooth, undetected ride straight into the nucleus.
Moving right along to Act 2, unmasking the hideout.
So what exactly happens after the virus gets inside the nucleus and integrates itself into our DNA? Well, modern antiretroviral therapy is an absolute miracle at stopping the virus from making new copies of itself. But the fugitive goes into deep hiding inside our DNA. And for the longest time, studying these hidden viral reservoirs was nearly impossible. To put it into perspective, old single-cell sequencing methods could only detect maybe 1 or 2 infected reservoir cells per patient, simply because the dormant virus wasn't producing enough recognizable material. But then, a team at the Gladstone Institutes developed this custom tool called HIV-seq. With this new technology, they successfully recovered over 1,000 reservoir cells from patients. This massive leap in detection suddenly allowed researchers to clearly see, literally for the very first time, what the virus is actually doing while it's supposedly dormant. And this brilliantly illustrates a really shocking biological transformation. Before a patient starts therapy, the infected cells are what researchers call fiery, meaning they are highly inflammatory, cytotoxic, and aggressively churning out the virus. But post-therapy, the virus actually actively manipulates its environment. The infected reservoir cells become quiet. They turn anti-inflammatory, and they activate specific pro-survival genes to actively evade cellular death. As senior investigator Dr. Nadia Rohn points out, the notion that the entirety of the HIV reservoir is latent is actually a misleading description because some reservoir cells can still be quite active. The hideout, it turns out, isn't completely silent at all. These supposedly quiet cells are actively ensuring their own long-term survival. They are flying right under the immune system's radar while continuously spitting out these small viral RNA fragments. The fugitive is quiet, for sure, but it is very much awake. And that explains exactly why patients on therapy still experience long-term inflammation.
Which brings us to Act 3, the unexpected alibi.
So we know exactly how the virus breaks in, and we know how it hides to survive. But how do we permanently lock it out? Well, remember that rare muscle disease we mentioned at the very beginning of this explainer? The one that seemed completely unrelated to an infectious virus? Well, we have finally arrived at the missing piece of evidence. It's time to solve that mystery. The disease is called LGMD1F, or limb-girdle muscular dystrophy 1F. It's this ultra-rare condition tracing back to a single family tree from Spain and Italy, and it's caused by a very specific genetic flaw, a deletion mutation in the TNPO3 gene. This mutation essentially adds a 15 amino acid extension to the resulting transport protein. Now, in the context of our detective story, this mutated TNPO3 protein acts like an impenetrable, deadbolted door. Let's move to the HIV cell cycle and see how this builds into the broader picture. Normally, the virus enters the cell, copies its RNA to DNA during reverse transcription, gets imported into the host's nucleus, and finally integrates by splicing itself into the host DNA. But researchers made a staggering discovery when looking at the blood cells of patients with that LGMD1F muscle disease. The virus could enter their cells, it could copy its DNA, it could even approach the nucleus. But it completely failed at the final integration step. The mutant TNPO3 protein was intercepting the fugitive right at the finish line. According to a really fascinating study published in PLOS Pathogens, this genetic flaw acts as an incredible natural genetic shield. Here's exactly how it works. The mutated TNPO3 protein forces another cellular protein, CPSF6. Actually, remember that host factor the viral capsid likes to bind to from earlier? Yeah, it forces it to relocate to the cell's cytosol instead of the nucleus. And this disruption is just catastrophic for the virus. It reduces the stability of the virus's pre-integration complexes by 3.1-fold. So the viral capsid basically falls apart way too early in the wrong part of the cell, heavily restricting the viral genome from ever integrating into the host's DNA. So the crucial point here is this specific number, 16. In cells carrying this specific muscle disease mutation, HIV proviral integration is reduced by a staggering 16-fold. This is absolutely huge because it's only the second genetic defect ever discovered in human history that provides this kind of strong natural resistance against HIV infection. Finally, let's wrap up with Section 4, Blueprints for a Cure. So why does tracking all these microscopic battles matter so much for us? Because right now, millions of people rely on lifelong therapy. Therapy suppresses the virus, but as we saw, it leaves a quiet, surviving reservoir that can rebound at literally any time. But by understanding the exact molecular lock on the CD4 receptor, by unmasking the surviving reservoir cells using the new HIV-seq tool, and by studying the natural resistance mechanisms found in a rare muscle disease, scientists are finally piecing together the ultimate blueprint to not just suppress the virus, but to eradicate it completely. It's truly amazing how interconnected human biology is. I mean, who'd have thought that mapping the shape-shifting disguises of a viral capsid and studying the genetics of a highly localized rare muscle condition would converge on the exact same microscopic battlefield? It leaves us with a really provocative thought to ponder today. Could the exact genetic flaw behind a debilitating rare muscle disease actually hold the ultimate key to finally eradicating HIV? It is a thrilling frontier in medicine. Thank you so much for joining me on this explainer. Keep learning, keep questioning, and I will see you next time.